Okay, so it's time for the second part of the video. So I hope you're excited, because I am, because this is a really cool mechanism. So in the first video, we discussed the HMP pathway and how that sugar is important, and we actually will convert it into PRPP, which is going to come a little bit later on in the pyrimidine synthesis mechanism, but I figured that we would just do that part first, so that way we can kind of just get right into it. So with the pyrimidine synthesis, it's quite complex. I tried to take out like enzymes that are not really, like, obviously we have the whole mechanism here, but I took out a couple of names of enzymes just because it kind of will make it a little bit cluttered. But just know that it is happening and the enzyme does exist. I just didn't put it on the board. So yeah, let's just get into it. So actually, this is kind of silly because I need to use the other whiteboard first. Look, one has wheels, one doesn't, so it's a little ridiculous here, but that's fine. Let me just make sure that you can see it. Oh yeah, that's perfect. Okay. So the pyrimidine, so the structure that we have here, this is our goal. So we're going to be putting aspartate, glutamine, and CO2 into this ring. This is like the base. And then we're going to be adding a sugar to it and phosphate. And so this is kind of the goal. So I figured if we can see where the goal is, then maybe we can get to this point, which hopefully we will. So yeah, so now I'm going to put this back. This is really silly because I should. Okay. So yeah, let's get into the mechanism here. All right. Let me just make sure that it's in the, yes, perfect. Okay, so this is the pyrimidine synthesis mechanism. We'll kind of just get right into it here. So the first thing that we have to do is we need to get, basically the goal for right now is just to get to UMP. So like I said in the beginning, we're going to have to put glutamine and CO2 into this. We use an enzyme called CPS2. There's a CPS1, which I talked about in the urea cycle video. That's located in the mitochondria. This is located in the cytosol. You put 2 ATP in, you're going to get 2 ADP out and 2 phosphate out, and also you're going to get a glutamate out. And so the goal of this enzyme is to create carbamoyl phosphate. And it does have some things that can activate it or inhibit it. An activator would be ATP or PRPP, which makes sense because we're going to be putting PRPP into this eventually. And then with this, where is it? Sorry, with the, yeah, and then with the ATP is going to be required to get this, this reaction going here. So yeah, so that's kind of it. And then UTP, this is technically feedback inhibition. Because, or product inhibition, we have UTP, which we create later on in the mechanism, and it's actually going to go ahead and inhibit this, which makes sense. It's basically going to say, hey, there's so much UTP, you should slow down a little bit. That's kind of cool. So yeah, so we have the carbamoyl phosphate. Once that's produced, we're going to hit it with some aspartate, which is the next thing that goes into the ring. So now we've covered the glutamine, the CO2, and the aspartate. Aspartate is going to go in, and it's actually going to do like a do-si-do type thing with the carbamoyl phosphate. So the phosphate is going to be swapped out for an aspartate. This is done using aspartate transcarbamylase. And then you're going to take water off of it, and it's going to become dihydroorotate. The next thing you're going to do is you're going to use a dehydrogenase, which is just a dihydroorotate dehydrogenase. You're going to hit it with an FMN. And then you're going to get an FMNH2 out of that, and you're going to create orotate or orotic acid. This is important, and I'm going to come right back to it. I just want to get through these next two steps. So the next thing, the OPRT, that is going to take this PRPP, which was created using the HMP pathway, and the PRPP synthetase, which we covered in the previous video, and it's going to add it here. So it's no longer going to be orotate, it's going to be called OMP. So that's kind of cool, and phosphate is going to come off of that in order to get this going. The next mechanism is actually going to remove a CO2, and then that's going to become, using an OMP decarboxylase, that's going to be turn it into UMP, which is really important. So basically what we're doing is we're kind of just swapping things around, doing some dehydrogenase reactions, removing some water. And this is important because orotic acid urea, that's an excess amount of orotate. This can be due to a deficiency in the OPRT enzyme or the OMP decarboxylase enzyme. And I believe from the notes, this is a multifunctional. I think that these two might be attached to each other. It's like an enzyme with two components to it. I believe that's what the note said. So basically, if this guy's deficient, well, that's not good news because you're going to get an excess amount of orotate or buildup. And that can have some deleterious effects on the body. So that's the clinical significance of this erotic aciduria. The UMP can then become a component of RNA, and that makes sense because if you think about the base pairs, uracil is going to be one of the base pairs for the pyrimidines. So that makes sense. You're going to have your sugar, you're going to have your phosphate on it, and then you're going to have your base. So right then and there, we just created our first ribonucleotide. So then we're going to just hit it with a kinase. We're going to add a phosphate to it, and then that's going to convert it into UDP. So then once we get to this point, I kind of consider it a branch point because once we get here, we can either go in two directions here. Ultimately, we're going to be making DNA, and then in this mechanism, we'll be creating RNA along the way. So we'll just kind of get into it. So this mechanism, which I think is kind of really interesting, so we use something called a ribonucleotide reductase. And I'm going to move the boards again. Whoa. Okay, let's see. Is that in the video? Yes, perfect. Okay, so ribonucleotide reductase. I'm not a very good drawer, so I just kind of did a simpler version because this is how I kind of think of it. But basically, you're going to have your sugar with some phosphates attached to it, and you're going to have an OH coming off of that second carbon on your sugar. If the sugar is a ring, and obviously you're going to have a base on it as well. And what this ribonucleotide reductase is going to do is it's literally going to remove that oxygen. I'm pretty sure, so it removes the water off of it. So it's going to take this oxygen off, and then it'll take off 2 more hydrogens with it. I didn't go into the specific mechanism for this, but I do think it's a very cool mechanism because you're literally going to go from OH to H. And that makes sense because this is ribonucleotide. This is deoxyribonucleotide. That's pretty cool. I thought that was fascinating because where the names came from and everything, it can be a little confusing, but if you think about it, it's losing this oxygen. So that's why we're calling it the deoxy. Okay, cool. So now I'm going to move the whiteboards again. This is kind of... Let's see, are we in the frame? Yes, okay, perfect. We'll just move over a little bit. Okay, so this is, we're going to be using a ribonucleotide reductase, which I just showed there. So we're going from the UDP form to the deoxy UDP form, or DUDP. So if you see here in this mechanism, we're going to be using the thiorodexin reductase. So we're going to be hitting it with an NADPH. That's going to get converted into an NAD+. And these H's are going to come off and create these I think they're called thiorodexin, I'm pretty sure. But I just kind of abbreviated it. So you're going to have 2 of these SH's. And what's going to happen is they're going to change into water, and then they're going to change into a disulfide bridge, which is pretty cool. And then this, it kind of will get reset using this thiorodexin reductase. So it's just going to kind of keep going through. It's a pretty cool mechanism, actually. But yes, you're going to get a water off of it. So that's the water that will come off and convert it literally from UDP to deoxy UDP. Cool, so that's a cool mechanism. Then we can take a phosphate off of it and turn it into dump. And then this is also another really cool mechanism, or at least I think it is. Maybe you might not think it is, but I do, so that's fine. So it's gonna go from dump to DTMP. And so basically what I use some abbreviations here. So FH4, that's the same as like a THF, tetrahydrofolic acid. So you have a N5, N10 methylene tetrahydrofolate. I just abbreviated it as FH4 because that's what I saw in the nomenclature. And what you're going to do is you're going to just convert it into dihydrofolate using the thymidylate synthase. And then we're going to have to reset this. So using I don't have the name of it. Oh, it's right here, sorry. So using this dihydrofolate reductase, we're just gonna do a similar mechanism to this. We're just going to, we're basically just gonna literally reduce. So the NADPH is gonna be converted into the NAD+. The clinical significance for this, so with mammals, MTX can actually halt this process. I believe in the human medicine, they use it to stop cancer from growing in different types of cells like that. But in a veterinary aspect, or I guess also human aspect, we can use something called trimethoprim, which will inhibit this process in bacteria. And I think some veterinarians will actually use, it's called SNZ. It combines a sulfonamide with trimethoprim, and it's just abbreviated SNZ. So that's like a clinical significance. That's why I think it's cool. And it can be inhibited using the 5-fluorosyl. And so this process creates DTMP, which then can become a part of DNA. Cool. So now we've done the U, we've done the T, and now we're going to have to do the next ones. And you may be asking yourself, well, why is there no TMP? And that's because there is no, for RNA, there's no, I believe, yeah, there is no TMP in it. It's not a thing, it's replaced with the U, or at least that was my understanding of it. So yeah, so we'll go back to this. So we have the UDP, you can hit it with a kinase and a phosphate, that'll convert it into UTP. This is kind of, I'm left-handed, so maybe I'll just stand on this side. UTP can be converted using a CTP synthase. We're gonna put some ATP into it, glutamine, and we're gonna get out a glutamate, an AMP, and a phosphate. So then once we do that, we're going to create CTP, and then this, I call it like another branch point, but you can remove 2 phosphates from it and create a CMP, which then can be used for RNA. And then CTP can also be converted into CDP if you just lose 1 phosphate. I should probably put that in. Cool. And then we're just going to do this same ribonucleotide reductase mechanism. And I don't think I mentioned the activator or the inhibitor, so this is the same mechanism, but instead we're going to be going from CDP to DCP. Sorry, DCDP. That's kind of a mouthful, hey? So yeah, so an activator for this would be ATP, and then inhibitor would be hydroxyurea or DATP. I believe that the hydroxyurea is also used as an anti-cancer drug, specifically with a chemotherapy type thing. I'm not sure if we use it in the veterinary world. That would be kind of maybe something I should have researched before I did this video. Yeah, I'm just gonna keep going. So the CDP can get converted into DCDP, and then from there we can just knock off a phosphate from it and turn it into DCMP, which then can be a part of DNA. So yeah, that's the pyrimidine synthesis mechanism. I'm gonna do another video on the purine synthesis. And yeah, if you have any questions or you found any errors with the video, please feel free to message me or leave it in the comments. I hope you enjoyed this video. Thank you.